Good morning, this is Dr. Rajesh Yadav from Department of Biochemistry. Today we will discuss about the fluid mosaic model. So in this slide we will discuss about the structure of the fluid mosaic models, the significance of the fluid mosaic model, role of cholesterol in the cell membrane, asymmetry of the membrane, and movement of the lipid in the cell membrane. So the fluid mosaic model was proposed in 1972 by Singer and Nicholson, and it was published in the most prestigious journal Science. So it revealed about the integral membrane protein. So the unique property of the cell membrane is the integral membrane protein, which is responsible for the movement of the ions. So fluid mosaic model explains various observations regarding the structure and function of the cell membrane. So protein and lipid is present in the cell membrane, and apart from the protein and lipid, carbohydrate is also present. The carbohydrate is present on the outer surface of the cell membrane, which is responsible for the recognition of the cell. So I would like to explain about the small clips of plasma membrane and fluid mosaic model. So with the help of this model, we can explain all the properties of the plasma membrane. So cell is made up of cell membrane. So it is covered by the cell membrane, and large cell organelles are present within the cells. The cell membrane is made up of phospholipid, and phospholipid has head and tail. Head is a polar which is made up of glycerol and phosphate, while tail is non-polar, it is made up of fatty acid. Because head is made up of glycerol and phosphate, it has polar nature and it is responsible as a hydrophilic head, or its nature is hydrophilic. While tail is made up of hydrocarbons, so it has hydrophobic nature. So these phospholipids form the cell membrane, and it gives the regular packing to the cell membranes. So tail is made up of hydrophilic tail, while head is a polar hydrophobic head. So these phospholipids are from regular packaging of the cell membrane. Integral protein is present in the cell membrane, which is responsible for the movement of the ions from outer surface to the inner surface. And it is also responsible for movement of the ion from inner surface to the outer surface. Carbohydrate is present on the upper surface of the cell membrane, that is called glycoprotein, and it is responsible for the cell recognition. Apart from the glycoprotein, the cholesterol is also present in the cell membrane, and cholesterol is responsible for the fluidity of the membrane that we will discuss in the next slide in detail. So in this way, the integral protein and cholesterol is present within the cell membrane. So with the help of this fluid mosaic model, we can explain a large number of the properties like fluidity, asymmetry, and all these things. So there is a modification of the cell membrane. Lipid raft is one of the another modification of the cell membrane, which is mainly made up of cholesterol and sphingolipid, and it is responsible for the cell signaling, transport of the lipid, and immune function. There are a large number of the function of the lipid raft, but we will discuss here only the small portion. So this is the structure of the lipid raft, which is showing here, that is present on the cell membrane, and it can move on the cell membrane. So it can transport the lipid inside the cell membrane. We will show here the structure of the saturated fatty acid and mixture of the saturated fatty acid and unsaturated fatty acid, which is present in the cell membrane. So what will be the effect of the saturated fatty acid and unsaturated fatty acid? Because if the saturated fatty acid is present in the cell membrane, they make the strong van der Waals interaction that will be responsible for the tight packing of the cell membrane. So it will provide the rigidity of the cell, while the mixture of the unsaturated fatty acid and saturated fatty acid, it provides the weak van der Waals forces in the cell membrane that will responsible for the fluidity of the membrane. So here we can see the upper structure that is showing here the paracrystalline state. When all the lipid bilayer, all the lipid molecule which have the regular or tight packing that form the paracrystalline state, and it provides the rigidity to the cells. While the lower figure is showing that irregular packing. So irregular packing provide the fluidity to the cell membrane, and cell have low amount of the rigidity. So rigidity or fluidity can be explained by this fluid Mosaic model. We can also see here the effect of the cholesterol on the fluidity. Cholesterol alter the phospholipid packaging and mobility. So if the cholesterol is not present in the cell membrane, so the cell membrane has very lower fluidity. So short unsaturated tail decrease the fluidity, while long and saturated tails, it disturb the packing and increase the fluidity. So when the cholesterol is present in the cell membrane, it will disturb the packaging and it provide the more fluidity to the cell membrane. So cholesterol is responsible for the fluidity of the cell membrane that can be explained by the fluid Mosaic model. So with the help of fluid Mosaic model, we can also understand the membrane asymmetry. In the cell membrane, the outer lipid bilayer is made up of phosphatidylcholine. You can see in the figure here, this is the phosphatidylcholine. So phosphatidylcholine is present on the outer leaflet, while phosphatidylethanolamine is present in the inner membrane of the cell membrane. So this is the phosphatidylcholine. It has cylindrical shape and it's present in the outer cell membrane, while this is a phosphatidylethanolamine. It is present in the inner leaflet and it has conical shape. So this presence of phosphatidylcholine and phosphatidylethanolamine provide the asymmetry of the cell membrane. Apart from the phosphatidylcholine and phosphatidylethanolamine, another molecule that is called phosphatidylserine. Phosphatidylserine is present in the inner surface of the cell membrane and that can flip in inner to the outer surface, and such type of movement is called flip-flop movement. So flip-flop movement is another important property of the fluid Mosaic model. Phosphatidylserine is a signal to the macrophage to clear the aged RBC. So many properties of the cell membrane that can be explained with the help of fluid Mosaic model. So here in this diagram we can see that membrane protein. So there are three types of the protein is present on the cell membrane: flippase, floppase and scramblase. Flippase protein is responsible for the movement of the lipid from outer surface to the inner surface, while floppase is responsible for movement of lipid from inner surface to the outer surface, and scramblase is responsible for the movement in the both directions. So in the basis of these proteins, and there are three types of the movement which are present in cell membrane. First one is a flip-flop movement. Flip-flop movement reveals the movement of the lipid molecule from one lipid bilayer to the another layer. That means this molecule will shift from inner layer to the outer layer or outer layer to the inner layer. That is, movement is called flip-flop movement. Another movement is lateral diffusion. In the lateral diffusion, the phospholipid molecule will diffuse from one position to the another position. So suppose this molecule will go here in the another, replace the another molecule. So this is the lateral diffusion. And rotational movement, all phospholipid is showing the rotational movement, that is the axial movement, or phospholipid is moving on its axis, that is a rotational movement. So three type of the movement is present in the plasma membrane that can be explained by the fluid Mosaic model. Thank you.